Thank you so much for having me. It's really great to be here. Yeah, and today I'm going to talk about, I guess, the way we think about structure-based de novo design, and specifically some of the work that we've done in this field over the last couple of years. I'm centering this really on some kind of quite exciting and unpublished work where we've been trying to apply these methods to the design of antibodies. So yeah, quite a lot of new stuff today, which is exciting. But so, at the Institute for Protein Design, we care about trying to design de novo proteins. And I think it's useful to ask kind of why do we want to do this, right? And simply put, nature has really only explored a tiny subset of the possible protein landscape, and importantly, evolution does not necessarily kind of select for the attributes of proteins that we care about in, say, a pharmaceutical or like a biotechnological perspective. Evolution doesn't necessarily select for in vitro solubility or stability or these things that we really want when we're making, proteins in kind of these biomedical or technological applications. So with de novo design, we kind of really hope to be able to derive new proteins with both new functions, but also kind of more desirable attributes, right? We want to be able to make kind of better proteins than those that nature has given us. And in lieu of a much longer introduction, I just want to tell you that the way that we kind of do de novo design typically follows this kind of four-stage pipeline. So, firstly, we try and design protein backbones, so structures that we think would be capable of carrying out or holding some particular function. And once we have these backbones, we try and find sequences that can encode those backbones. We try and find sequences that, when expressed, would fold to that particular structure and would have the function that we desire. And typically, we can design many, many more sequences than we're able to test experimentally. So we need really, really good computational filters to, separate the good designs from the bad designs. And then finally, we take those designs that we think have a good chance of working into the lab, and we express these and experimentally test whether or not they have the function that we desire. And for many of the applications that we really care about, the big limiting factor a couple of years ago was our ability to make protein backbones. We were limited by our ability to make structures that would be able to carry out some function. We got quite good at these other steps, but we were really limited for a huge number of applications in our ability to make these protein backbones. And this was really the work of my postdoc, was trying to develop sort of general methods to improve our abilities to do this step alongside many, many really excellent collaborators. But the work I'm going to tell you about today is about a neural network that we trained called RFdiffusion, which kind of built directly on advances in structure prediction that Serena has mentioned in the previous talk, but also then in advances in kind of generative models for other areas such as image generation. And specifically, we turned to this sort of class of generative model called diffusion models. And I think this is a really nice schematic for kind of like demonstrating how diffusion models work. The basic kind of principle is that you have some data or like a data distribution. So in this case, this is like images of dogs. And you can define some corruption process or noising process such that you can kind of iteratively add noise to this data, right? So that, halfway through, you have these noisy images, which still clearly bear some dependence on your starting data. But with sufficient additions of noise, you converge to a kind of random distribution. So typically we use Gaussian noise. Many Gaussians drives you towards a random Gaussian distribution. And then what you can do is that if you train a neural network to undo each of these steps, you can basically now sample from your known random distribution. So you can sample from that Gaussian distribution. You can feed that into your neural network. And then over all of these kind of iterative steps of denoising, your neural network will now land you at data which very closely matches the distribution of data that you trained on. So it can now generate new images of, dogs in this case. So diffusion models have really been shown to be state of the art. These are what underpin kind of DALL-E and these models that many of us will be familiar with. And we kind of realized, and we weren't the first people to realize this, but we kind of recognized that diffusion models had a number of characteristics that lent them really nicely to protein design, that would make them a really good fit for the kind of things that we want to do in protein design. So, firstly, because you have this, you're sampling from this random distribution, and actually at each step of denoising, you are adding further noise, further randomness. And what this means is that you can generate essentially endlessly diverse outputs with these models. So you can sample as many things as you like. And this is amazing, right? Because this gives you many, many shots on goal. If you're trying to do a hard problem, it's nice to have many different starting points. At least in the way that we formulated this, we can also operate directly on amino acid coordinates. So we're building protein structures, but we're building them from the actual backbone amino acid backbones, essentially. So we can operate directly on the coordinates themselves, which is very handy. And then finally, we don't normally care about just making any protein. We want to make proteins with specific functions. And the nice thing about diffusion models is that there are many ways of doing this kind of conditional generation, right? You can either kind of guide these with kind of these auxiliary potentials, or you can just train conditional models. So this is really convenient. And we kind of recognize that diffusion models might be the answer to this kind of first step of the protein design pipeline. And we therefore kind of set out to train a diffusion model for protein backbones. So the principle is the same, right? You start from known data. So this is protein structures in the protein data bank. And you can now define some corruption process which drives you from protein structures to random Gaussian distributions, essentially, and uniform distributions of rotations. And you can then essentially train a neural network to reverse that process such that now you can sample from that known distribution and now hopefully generate new proteins. So this is the kind of principle, and this is what it looks like when we actually train this. So, you can basically see at the start of this that you have these kind of balls of random amino acid frames, and then these get denoised to generate, in this case, a big symmetric oligomer. So this was RFdiffusion. We trained this neural network. I'm not going to go into all of the details about this now. This is now, this work has been out for a year or so now, so people can read about this if they're more interested in the details. But just to say that we were able to really apply this to a whole range of different problems. We were able to show that this could, for many of the problems that we cared about in protein design, be this kind of first backbone generation step. Yeah, so it was kind of met this kind of broadly applicable criteria. But the one thing I am going to focus on here is this bottom left one, this binder design problem. And, when we do binder design in RFdiffusion, essentially what we're doing here is that we're saying, here's a target protein, make me a protein that binds to this. And this has been a kind of like quite long-standing challenge in protein design, right? And it's because, being able to design binders is really phenomenally useful. When we're thinking about protein structures, if you can design a binder to interact with a very specific epitope on a target protein, you have like real control over the function of that, right? If you can target, say, a specific conformational state, you might be able to induce agonism or antagonism. And that only works because you know the structure, right? You know where you're targeting. You might bring together two things and create kind of bispecific functionality. There's a ton of things you can do if you can design binders. And importantly, because you can define the epitope, you can also then decide whether you get sort of a broadly neutralizing binder if you're trying to target, say, a virus, or if you can hit a very kind of a very small mutation, for example, if you're trying to hit, say, a cancer antigen or something like that. So we really want to be able to design binders. And, in the paper, we were able to show that RFdiffusion really excels at this task. So, now it's quite a simple process to make a binder. You can basically take a target protein, you can specify a few residues that you want to interact with, and you can now do this denoising process, and you can, sample noise and then generate nice sort of shape complementary binders that interact with that target protein, and you can then find sequences that encode them. And what we find is that these work really nicely in the lab. So in the paper, we were able to design binders to 5 different targets. And the really cool thing about this is that we were just able to do this with dramatically higher success rates than we were previously able to. So when we compare sort of our success rates on these same antigens versus the kind of original binder design pipeline, you can see that we're, in many cases, 2 orders of magnitude higher efficiency at creating binders now than we used to be. And what this means is that we can now do much smaller scale and faster experiments to generate binders. So we can actually, get binders much, much faster than we were previously able to. And, since this work, there have been 40 or 50 cases internally of successful binder design campaigns, including to targets that we weren't able to get binders to previously. So that's the kind of really exciting thing about what new technology can bring you. And this is just one of the structures that we've solved now. And, invariably, when you get these binders, they bind as you design. So this is just showing a cryo-EM structure of the design model shown in purple and the cryo-EM structure shown in gray. And you can see that, they're essentially indistinguishable. This binds exactly as we designed it to bind. So, that was RFdiffusion. The paper is published. It has a very detailed supplement if people are interested in the details and more of the data. And the code is also completely freely available. It's on GitHub. There's also a CoLabFold that makes this really kind of easy to run. So yeah, do check it out if you are kind of keen to try and integrate these things into your research. But the stuff I really want to talk about today is, moving on from this. So, I've told you that binders are really good and important. And actually, many people have worked on de novo binder design. So originally, this was cracked with a kind of Rosetta-based pipeline. We then made advances. Bruno Correa's lab have this massive method for designing protein binders. And what you can kind of realize from looking at these is that all of these methods typically rely on making very kind of rigid interfaces with the target epitope, right? They bind predominantly through helices, sometimes through strands, but with these highly ordered and highly rigid interfaces. And the kind of interesting thing is that that's not how nature does it, right? When we think about what nature does, the way that nature has solved the binder design problem is through antibodies, right? Combinatorial shuffling of CDR loops, right? So they make loop-based interactions, which are, in the antibodies case, able to interact with, most epitopes that your body will encounter. And this is inherently more challenging, right? Because antibodies are binding through these flexible loops, and no one to date really has cracked how you design these sort of much more flexible loop-based interactions. And that's how kind of essentially all immune systems have done this, both in antibodies and nanobodies in camelids and sharks, and then you have these kind of synthetic versions. But fundamentally, these are all binding through loop-based interactions. And we kind of wondered, coming out of the RFdiffusion project, could we actually train a diffusion model to do de novo antibody design? This is a kind of, properly unsolved problem, but, with collaborators at the IPD, we wondered, maybe now is the time to try it. So with Nate Bennett, who's really co-led the computational side of this project, and then Rob Rigot, who's led the experimental side, and Andrew, who's been doing the cryo-EM, we really set out to do this, along with a number of amazing collaborators on this project. And so, what we'd learned from RFdiffusion is that, this kind of way of training generative models yields models which can generate very high quality and also diverse protein interfaces, right? And although antibodies bind through these loops, fundamentally, it's the same thermodynamics that govern these interfaces. So we kind of thought, can we apply RFdiffusion to do antibody design now? So in other words, can we, train a diffusion model whereby we sample random noise, and then the model kind of progressively denoises this to generate an antibody that would interact with the specified epitope. So you would provide a target and some specification in orange of where you want to design an antibody to bind, and then you would get an antibody that interacts with that. And I made the world's worst schematic earlier today, but basically just to clarify what we're doing here is that we want to be able to fix the framework. So we want to be able to say, this is an IgG, or this is a Fab, or a nanobody, or whatever, and we're fixing that part of the framework in sequence and structure. But then we want to be able to design de novo CDR loops, so loop-based interactions to target a specific epitope. And so what we did is that we took RFdiffusion and some kind of underlying improvements to the model, and then we trained this on kind of relevant data sets. So we trained this on antibodies in the PDB, TCRs, any interaction that we could pass from the PDB that was kind of loop-based in the way that it interacted. We reasoned that, learning as much as possible as you can about loop-based interactions would help this model. And then such that we can specify the antibody framework, we provide the sequence and this template representation of that to RFdiffusion. So how that looks is something like this. So if we want to design a nanobody, for example, we can basically represent a nanobody three-dimensional structure as this pairwise representation. So this is basically like a every pair of distances and angles between each residue. So this ends up as a two-dimensional matrix. We, of course, mask the CDR loops because the model is designing them. We don't want to give them to the model. But we can basically now represent any antibody framework with a sequence of the framework and this two-dimensional kind of pairwise representation. And then this is the architecture of RFdiffusion, which I'm not really going into today, but just to say that we can then provide these inputs to RFdiffusion such that we can now at design time specify precisely which antibody framework we use. So yeah, this pairwise goes in the pairwise track of RFdiffusion, and the sequence is provided to the model. And what this means is that we can now, sample Gaussian noise, and we can say, well, I want to make an antibody now, or I want to make an scFv, and RFdiffusion is capable of doing exactly that. So we can design, an antibody and an scFv, both of which are interacting with this target epitope. Another really nice thing we want to be able to do is, although we care about where the antibody is binding, we don't really care about the rigid body orientation of the antibody to the target, right? We want that to be sampled by the model, so long as it's, finding different ways to interact with that same epitope. And the really nice thing about these pairwise representations is that they don't tell the model anything about how the antibody interacts with the target in kind of rigid body space. So what that means is that you can sample, your Gaussian noise and run the model 2 or 3 times, and you will get diverse docks, right? So you can see that these 2 antibodies are both interacting with this epitope, but with very different kind of docking modes. So this is a real feature, right? We can now sample like tons and tons of different antibody docks, which all interact with the epitope that we want, which is pretty cool. And then finally, to get that epitope targeting, the way that we actually do this is that, during training, what we can do is that we can look at an antibody structure, and we can work out which residues on the target, that's shown in orange, are interacting with the CDR loops, right? So we can say which residues on the target are via CDR loop. We can pass that out and then just essentially provide that to RFdiffusion. And typically, because we don't want to have to specify the full interface, right, at design time, that's super annoying. So we just mask this heavily during training. So the model kind of learns to respect these 2 or 3 residues that we provide I have to interact with CDR loops. So this is really nice, and this works very, very cleanly, really. So you can now, for example, take a target, and you might have 2 epitopes on this that you're interested in. If you provide the kind of orange residues as input, you get an antibody that binds there. And likewise, if you provide the pink ones, you will get an antibody that binds there. So we have kind of full control over the antibody framework and the epitope to which antibodies bind. And so the question, I've just shown you pictures at this point, and the real sort of proof of the pudding is whether this actually works, right? So we set about to try and experimentally characterize this. And we started with these single domain antibodies, so nanobodies, because they're much easier to test experimentally. But we designed lots of nanobody binders against a whole range of different targets. And really nicely, we see that we get binders to most of the targets that we design against. So we have nanobody binders to RSV site 3, to COVID RBD, to flu hemagglutinin. We can get these binders. And these are somewhat modest affinity, which I'll come back to. But the key question is, are they binding where we design them to, or is this just some non-specific binding between a poorly behaved protein and a target? So for that, we can ask, well, can we actually do competition experiments? In the case of this COVID RBD nanobody, we actually have a de novo binder, structurally characterized de novo binder to that site, right, which should be able to outcompete the binding of that nanobody if the nanobody is binding to the correct epitope. And indeed, that's exactly what we see. So if we add the de novo binder, this mini binder alone, you get kind of no signal. When you add the nanobody, you get a big jump in the signal, but then nanobody plus mini binder completely abolishes that. So in other words, the nanobody is binding to the epitope that we designed it to. And we can show the same thing for this TCDB. This is a toxin from C. difficile, which is another target that we were able to get nanobodies to. And you can get these kind of 300 nanomolar nanobodies, and again, they perfectly compete, right? The mini binder plus the nanobody completely abolishes binding, which is very cool. So we know that we have, on-target binding here. And I just want to talk a bit about the affinities here, right? So, we have got used in our field a bit now to expecting kind of low nanomolar affinity binders straight off the computer. And that's not really what we've seen. We've seen much more modest affinities, right? Like hundreds of nanomolar to low micromolar. But when we compare to kind of the early de novo binders, these early mini binders, when this problem was first kind of cracked, actually our affinities are really similar to what they had coming off the computer, right? So, they're hundreds of nanomolar to low micromolar. And we fully expect to be able to do what was done then, which is to experimentally optimize these, right? We can just do directed evolution or, affinity maturation, whatever you like, to get to higher affinities. So we're not really too worried about the modest affinities that we see at the moment, because we know that we are binding to the epitope that we have targeted. And what's, another question is, are we just copying what's in the PDB, right? Are we just copying what we see in nature already? What was really encouraging to see is that no, we're not. I think so. This is shown in pink is our designed nanobodies. These are 4 nanobodies binding to flu hemagglutinin, aligned on their kind of closest nanobody or Fab that we can find in the PDB. And you can kind of see that they're not interacting in the same way, and they're kind of highly distinct from what we see in the PDB. In the case of this TCDB toxin, there's actually no nanobody or antibody binding to this site that we designed against. So yeah, again, this is like really not copying from the training set. Which is encouraging. It's always a fear when you're fitting models on the training set that you might memorize these things. And we can also look at how similar we are at the sequence level on the CDR loops to things in the training set. And again, we find that, we're not that similar, right? We obviously show some similarity to CDR loops. These are relatively conserved in some positions. But, overall, there's no sign that we're kind of memorizing and regurgitating the training set. And also, of the ones that work experimentally, it doesn't seem that these are more similar to the training set than ones that don't work. So we're really confident here that we are doing, properly de novo design of antibodies and nanobodies, and that this is generalizing beyond what we see in nature. So then really the final question is, how accurate are these? I've told you that they're binding to the correct epitope, but that doesn't mean that they're necessarily, totally structurally accurate. So here we, turn to cryo-EM to try and resolve this question, right? So we took a nanobody to flu hemagglutinin, and you can see, it's a bit hard to see at the top, but you can see in these class averages that you get really nice particles here. And when we do a 3D reconstruction, you can already see the nanobody bound here, right? You don't have perfect occupancy, which I'll come back to, but you can see the nanobody binding to the stem of this flu hemagglutinin. And when we solve the kind of high resolution cryo-EM structure here, you can really see that the nanobody is binding almost exactly as we designed it to. You can see that it's kind of pushing up this glycan in teal, and the overlay of these design model and cryo-EM structure is really, really close. It's binding exactly kind of in the mode that we designed it to. And when you look at this CDR-H3 loop, you can see that, it's really extremely accurate. And this is just showing the overlay here of that. So we're about 0.8 angstroms off on the CDR-H3, which, given that this is the longest of the loops and the most flexible and hardest to model of loops in antibodies, we were really, really excited to see this. And you can actually look at the side chains and see that the rotamers are really kind of almost in the perfect position, which was just incredibly exciting. Fun thing to note about why we don't see full occupancy, we didn't notice that there was a big glycan here in the structure. So actually it's quite cool when you look at the unbound protemer and the flu hemagglutinin, you can see this glycan is occluding the binding site, but when we get the nanobody bound, you can really see it like pushing up the glycan. So yeah, a mistake, but it's quite cool to resolve that by cryo-EM. Yeah, so just to conclude, really, I've told you that the published and publicly available RFdiffusion version is kind of broadly applicable and could be used in, I think, many, many design scenarios. And that now, I've shown you that we've extended this to design antibodies and nanobodies, which we hope will really be the kind of basis of huge pharmaceutical futures. Now, if you can get to a point where you can just specify an epitope and you can now design an antibody with all of the nice properties of antibodies that we know and kind of love, if you can get to that, then that's kind of an extremely attractive position to be in. And finally, I've sort of told you that we have sort of overcome this barrier of atomically accurate design of loop-based interactions. This ability to model loops accurately enough to design interfaces has really been sort of shown in this work. So yeah, with that, I will end and just say a thank you to everyone who's been involved in this project, especially Nate Bennett and Rob Regott and Andrew, who have really co-led this project with me, and then David Baker for being a fantastic mentor. And yeah, I would love to take questions if people have them.
Thank you, Joe. This is really great to hear about this work. While people get their questions, I will make use of my privilege of hosting. I will make you a question. So from the RFdiffusion paper, what I get is that mostly you design a backbone based on what you want, right? You select your epitope and you design a backbone with diffuse by diffusion.
Yeah.
And then you use methods like protein MPNN to design sequences that would fold into that backbone, right?
Yeah.
So when you moved into the new work of antibodies, my question is, because I did some studies and I get maybe the hints that sometimes MPNN it generates geometrically compatible backbones between two things, but not necessarily energetically optimized. So have you thought about including a final step of energetically refinement of the designs? Because maybe these low affinities is because, you know, they match geometrically speaking, but then the energy maybe is not optimized. Have you thought about this to improve it or to include some sort of this kind of strategies?
Yeah, totally. I mean, I completely agree that I think the use of protein MPNN here, which we do use in the antibody study as well, we use this completely out of the box, is like probably far from optimal. We have not really explored this step to date. I think, yeah, it's quite possible that either kind of Rosetta based or some other method for really trying to optimize those interfaces may well help. Yeah, I think that sequence design step at the moment we have really not tackled. We've only been working on the kind of this design step and to some extent the filtering step, but this yeah, this sequence design step, there's a huge amount of work to do in there.
Do we have questions from the audience?
I have something to ask in case no one else has. So I yesterday came across this paper from Baker Lab about RF diffusion model to model protein small molecules and others. So how is that technique different from this, or is it based on the same concepts? That's the first thing. And the second thing that I would want to ask is, like, in case you say you predict a lot of cases, what is the percentage of correctly predicted, experimentally correctly predicted cases that you get out of n cases that you predicted? So these are the two things that I wanted to ask.
Yeah, so indeed, the kind of this RF diffusion all atom is now out. So the concept is very, very similar. Yeah. So in something I didn't really talk about today is that RF diffusion builds directly on top of RoseTTAFold. So we actually take a structure prediction network, which has trained for like thousands of GPU hours, and then we fit a generative model on top of that. So this allows us to essentially train big generative models very quickly. But RoseTTAFold only models protein atoms, right? It has no knowledge of DNA or small molecules, these kind of things. So Rohit Krishna and Ju Wang in the Baker Lab essentially extended RoseTTAFold to model everything, right? So now RoseTTAFold can predict the structure of proteins with small molecules, with DNA, RNA, whatever you like. And then, once you have that model, you can essentially apply the same principles that we applied in the original RF diffusion paper. So Woody Ahern was able to then take that all-atom RoseTTAFold model and then fit a generative model on top of that, such that you can now do protein design in the presence of, say, a small molecule or DNA or something. So it's conceptually very similar on, exactly how this is done, but it allows a whole load of kind of new questions to be answered. On the success rate question, yeah, this is a great question. I think we have, at the moment, approximately like a 1 to 5 or 6% success rate. So yeah, I think we'd like to push that to the point that we're at with kind of de novo binder design, where we can often get 10, 20, 30% success rates. But at the moment, compared to kind of screening random libraries where you might have 1 in 10 to the 7 or 10 to the 8 or something, we're doing pretty well. So.
We do have a question from the chat. How did you design the CDR sequences on top of the RFdiffusion backbone? Also with protein MPNN or with something antibody specific?
Yeah, so at the moment we just use protein MPNN. Sorry, I moved slides. Yeah, we just use protein MPNN. I think the sort of challenge and what I wanted to show, although it's hard to get to the slide if I go all the way back to the beginning, is that, something which I haven't really spoken about today, although we've been doing a lot of work on, is, building out this whole pipeline. Right? This pipeline works really well. If you can, design backbones, design sequences, and know on the computer which of those are good, then you're in a really nice position, right? Because now you can, like, if you have a really good computational filter, you can kind of like know that you're getting better at these steps without having to do experiments. We've done a huge amount of work building on this and have had some success. But I think we're not as good at this yet as we would like to be. And because of that, because we can't reliably optimize these upstream steps without doing experiments, we tried to kind of fix as many components as we could, right? So we just thought, let's not touch protein MPNN. We know that protein MPNN is pretty great at designing sequences. We assume that will translate to the CDRs, so let's just fix that. But in the future, if we have better reliable or more reliable filters or just more experiments, more rapid feedback, we would definitely like to explore that. Thinking about trying to push for more kind of native CDR-like sequences or just sequences that might have higher affinity, this kind of thing. I think that's a whole step that we have not properly explored yet.
Thank you. We have another question from the chat. I guess we have room maybe for one extra if the answer is not too long. So from Ulisse, thank you for your presentation. Could you give us some insights into the applicability of RFdiffusion for enzyme engineering and main challenges in such cases?
Yeah, totally. So I think here we in the Baker Lab often think about kind of properly de novo design. So when we think about enzyme design, we think about designing kind of properly de novo enzymes that are not related to natural ones. In this case, extending RFdiffusion to model everything is really convenient because now you can say take a substrate, you might position side chains such that you're trying to stabilize that transition state, and then you can try and build a protein around that essentially. So this is how I would pose that problem. You would position side chains such that they would hope to stabilize the transition state, and then you build a protein around that. This is still a very hard problem because, of course, that is not really modeling anything about the kind of kinetics that you require in an enzyme. On the flip side, kind of in optimization of existing enzymes, we've had some success with some of these models, just using them to essentially idealize what nature has already given us. So Kira Samida in the lab did some really nice work where she basically took existing enzymes and replaced bits of the kind of all of the non-idealities on them, some of these long loops or poorly packed regions. She built kind of de novo protein to replace that, but didn't touch the active site, right? So just like left the active site as is and kind of improved the rest of the protein. And this then, can give you kind of still active enzymes, but that are much better behaved in the lab. And I think that's another really viable strategy when you have an enzyme that's already working.
So there's a person who says, thank you very much for your presentation and time. And the last one, so we close with this. There aren't many instances at around 8K of antibodies and antigen in complex fine-tuning a model such as RFdiffusion, which sounds like what's happening here. How do you improve that?
Yeah, this is a great question. Indeed, I wish we had more antibody structures. That we are in a data limited regime here. I guess I briefly mentioned this, but when we train RFdiffusion on antibodies, we do try and augment that data as much as possible, right? So we include TCR structures, and because the TCR MHC dock is so much more constrained than antibodies, AlphaFold is actually quite good at predicting this. So Phil Bradley at the Fred Hutch in Seattle generated a huge set of predicted structures of TCR MHC complexes, which we know are pretty accurate. So we train on them as well. So that gives us about another 2,500 structures. And then we can pass out kind of any interface in the PDB that is primarily mediated by loops. So this gives us about another 15 or 16,000, I think, structures. So yeah, that's how we generate our data set. And then so this is, I mentioned just earlier that we fine tune off of RoseTTAFold when we do this. I do think that starting from a big model that already knows a lot about proteins is definitely giving us a nice inductive bias here, right? That even though you only have a small data set to do this generative fine tuning on, the model has already learned a huge amount about what it is to be a protein and maybe an interface. So I think it is probably this fine tuning regime which is helping us as well model complex interfaces from a pretty small data set.
Okay. Thank you very much, Joe. We thank again Serena and Joe for this great two talks about this great topic of protein design. Remember that we are aiming at having these webinars every second Tuesday of each month. Sometimes we vary a little bit, but keep tuned to know about the next editions. So thank you all for attending, and see you soon.